Hello, everyone, and welcome to angelMD's Syndicate presentation for Salarius Pharmaceuticals. On the webinar with us today is one of our Syndicate leads, Dan Parsley from the Houston area, along with Salarius CEO David Arthur and several members of his team. Our other Syndicate leads on this deal are Dr. Fred Duffy, a plastic surgeon, and Steve Cooley, both of whom are from the Dallas area. Salarius is a Texas-based and CPRIT award-winning company developing epigenetic therapies for rare pediatric and genetically well-defined solid tumors. For more information on Salarius, please visit their profile page here on angelMD, as well as what is available on their Syndicate page. Dan, I'll let you take it from here to give a brief introduction on how you became involved with Salarius.
Great, thank you, John. It's a pleasure to really provide a testimonial, if you will, as to my interest in serving as a Syndicate lead for this very exciting and very important company, Salarius Pharmaceuticals. I like to kind of break down my assessment of them in three areas, and you'll hear more detail of each of these as David makes his more detailed presentation. But one is just the impact of Salarius addressing the currently incurable Ewing sarcoma cancer. Affects a number of children. This needs to be resolved and solved, and I think they appear to have a novel approach that just may do that. In addition, a much broader impact on the hormonal cancers that could have an extremely wide impact well beyond the Ewing sarcoma. Also, just looking at the validation of this company, and this is one thing that really, in our effort to begin to look at this more from an investment standpoint, the de-risking nature of this investment by looking at the institutions that have not only thrown their support but their monetary support behind it. Three notable being the Texas CPRIT grant program. We view them as having a very high bar, and Salarius is one of their larger grant recipients. Two is the National Pediatric Cancer Foundation. Clearly that speaks volumes about the importance of this disease of Ewing sarcoma, and in their encouragement of the early indications of Salarius. Three is the fact they're a Houston J-Labs company. They're one of the first resident companies here in Houston. Again, we view J-Labs as a premier incubator and a high bar for their selection process. And they've had a tremendous investment support, widespread support from perhaps as many angel groups and biotech life science focused angel groups that I've seen in my angel investment experience all across the nation, on the West Coast, the East Coast, and very significantly across the state of Texas. So those are all good validations that should give us confidence that there's a lot of very smart people who have been tracking this company and are putting good money behind it. And of course, we have to look at the investment potential of this company. And I think being an oncology therapeutic, focusing on an orphan disease such as Ewing sarcoma and some of the fast-tracking benefits of that, but also a very encouraging, compassionate use study that David can elaborate on with a young man who had this disease. He experienced some real positive response to it while he was taking the disease at the end of his life, and that certainly is a data point of one, I realize, but it's an encouraging one, and hopefully they'll see that kind of results from their phase 1 human trial. So I'm encouraged, and I'm putting my personal investment behind this, and hope to introduce it to more investors in my circle. So David, I'll turn it over to you.
Well, Dan and John, thank you for hosting us today. This is David Arthur, CEO of Salarius Pharmaceuticals, and joining me for today's presentation and Q&A is Dr. Daniela Santiesteban in Business Development and Scott Jordan, Chief Financial Officer. So with those introductions made, let's jump in and talk a little bit about Salarius. Salarius focuses on epigenetics, and this is the way the body reads your genes. So in an epigenetic illness, the body looks at healthy genes and misreads them. And many times what that misreading leads to two proteins being connected that should never be connected. And that connection is called a translocation. That translocation leads to what's called the creation of an oncoprotein, which drives cancer. Our lead compound, cyclodemstat, specifically targets this communication process and these translocations, these oncoproteins, by inhibiting the lysine-specific demethylase 1 enzyme, or LSD1. Now, what's important to know is, by targeting LSD1, we're targeting the tumor cells and not healthy cells. That's a key differentiation in our approach versus medicines we're all familiar with, chemotoxic agents. Now, LSD1 is a validated target in many solid tumors, and Danielle is going to talk a little bit more about that later. But what's really important to recognize is that in our early in vitro models, looking at cell lines in our early animal studies, we've shown that we can reverse the gene expression of certain cancers, and we've been able to cure cancers in certain animal models. Our technology has come out of the University of Utah. Our composition of matter patent has been issued in the United States and has been allowed in almost every major country around the world already, including China. And where it's not approved, it's still working through the process. Our composition of matter patent is the strongest patent you can have in pharmaceuticals. So that's the type of patent protection you look for when you're making a biotechnology investment. It's good through 2032 in the U.S., and that's without any potential extensions. We have a very seasoned management team that's delivered the company to this point. We'll talk about them in a moment. But from an investor perspective, I want to focus, as we go through this discussion, on compelling science, intellectual property, which we've just touched on, a capable management team, but most importantly, the financial opportunity that Dan touched on, and that is the ability to invest in a clinical stage biotechnology company that's receiving $21 million moving forward in non-dilutive capital. This results in a 4-to-1 investor leverage. Now, we're a widely validated target. Let's talk about the science. LSD1 plays in a critical role in proliferation, differentiation, cell migration, other areas of oncology. But what's very important to note is, while multiple big pharma companies are pursuing LSD1, we're going to talk about why Salarius is highly differentiated from its competition. We work better. We work in more indications, and it's not by luck, that's by design. We are a reversible inhibitor, not an irreversible inhibitor, and that means we bind in a slightly different location on the LSD1 enzyme, and that's what creates our competitive advantage. Now, this allows us to target markets with a high unmet need. Our initial strategy is to go after Ewing sarcoma, a rare, devastating pediatric bone cancer that the FDA provides numerous programs to help us succeed. We're going to quickly follow up in related sarcomas. And when I say related sarcomas, what I'm talking about is sarcomas that share the same biology as Ewing sarcoma. So back to that translocation, where two proteins that shouldn't be connected are connected. When these sarcomas share a similar half of one of those proteins, that means they have related biology. And by working in Ewing's, we should work in those related sarcomas, and we have animal data to show that we will. Turns out, LSD1 is an important enzyme in late-stage hormonal cancers, prostate, breast, and ovarian. There's some very exciting data there, and we look forward to entering those clinical studies at the end of 2018. So, speed to market, Ewing sarcoma, proof of concept, much larger financial opportunity in the larger, longer-term hormonal cancers. Now, let's just touch on Ewing sarcoma, our initial target, before turning it over to Daniela to go a little deeper in the science. Devastating cancer, Ewing sarcoma. It's rare, it's orphan, it's a pediatric illness. The average age of diagnosis is probably about 12 years old. The only available treatment approved is the use of adult chemotherapy, radiation, and surgical resection. Unfortunately, for 70% of the patients initially diagnosed with localized tumors that have not spread, these devastating current therapies don't work in about 30% of the cases. If you're unlucky and you're not diagnosed until the cancer is spread, you have a 90% 5-year mortality rate. You put all of those numbers together, and about 45% of the patients with Ewing's fail to respond to therapy, or they relapse, and they have a 90% 5-year mortality rate. And unfortunately, for the 60-65% of the patients that do respond, they're being treated, these are kids being treated with chemotherapy, radiation, resection, and they're just tremendous long-term physical and mental developmental issues. We think we can do something about that. We've shown cures in animals, and going back to my very first slide, we target only the cancer cells, not the healthy cells. We're not a chemotoxic agent. And to move us along quickly, the FDA has incentives that we are already taking advantage of. A priority review voucher, which I'll go to in the Q&A if there's interest, but upon approval for sale in the U.S., we are already pre-approved by the FDA to receive a voucher that we can then turn around and sell for $125 to $150 million. We've already been granted orphan drug status by the FDA, which provides us with additional protection from generics, and also puts us in a class of drugs that allows higher pricing. As soon as we see the results we're expecting to see in our phase 1 Ewing study, we will talk to the FDA about breakthrough status, which opens a new category of programs, all designed to move us along and potentially have this drug on the market in the U.S. treating patients in 2021. Now, at that point, we expect to have sold the company, but again, speed to market with Ewing sarcoma. Our IND is activated, and we are enrolling patients in 2018. Let me just touch on the financing for a moment. We are raising $28 million. We have already lined up $21 million in non-dilutive funding. As Dan touched on, that comes from the Cancer Research and Prevention Institute of Texas, where we're an awardee of almost $19 million. We have $14 million remaining. We are receiving funding from the National Pediatric Cancer Foundation, which is funding approximately half of our Ewing's clinical study. And the University of Utah is sponsoring our breast and ovarian study. You put all that together, and a $7 million equity raise actually gives us the ability to drive $28 million in value creation for investors. That gives us 5 shots on goal: Ewing sarcoma, 2 types of breast cancer, late-stage ovarian, and late-stage prostate cancer. All of this is designed to show proof of concept and set us up for an exit in 2020, first half of 2020, with a base case valuation of $400 million. Now, that's just Ewing sarcoma. That doesn't count any upside related to other sarcomas or any of the hormonal cancers. So remember, compelling science, intellectual property, management team to deliver, and the financial opportunity. I'm incredibly fortunate to have recruited a very seasoned management team. I won't go into the details. This slide will be available. But I will suffice it to say that in every key function across the company, I've been able to identify an individual with 20 to 30 years or more experience in their area of expertise, along with significant experience in building and exiting pharmaceutical companies, including both big pharma and small pharma. I'll suffice it to say I have yet to come across a problem that someone on this team has not previously addressed and knows how to solve. Now with that, I'm going to turn it over to Daniela, who's going to walk us through the science, and will be available at the end of the presentation to answer any questions you may have. Daniela, take it away.
Great, thank you, David. So I'm just going to give a little bit of background on epigenetics. If we think of a healthy cell as this healthy plan where all the different branches are controlling a different cell function, such as proliferation, apoptosis, differentiation. What happens in cancer is that some of these branches start to grow wildly out of control. What a lot of the other cancer therapeutics do is they specifically target that branch that is growing out of control. But if you've ever done any yard work, you know that if you have a weed in your lawn, in order to get rid of that weed, you have to target that weed at its roots. So what happens in a lot of these cancer treatments is that they work for a little bit, but then those cancer cells develop resistance to the treatment, and they're harder and more difficult to treat. The Salarius approach is to target the problem at its roots. By doing this, we have better control over that cell's fate. We have less of a chance for that cell to develop resistance. And we have a lot of data that shows that we work very well at completely reversing the cancer phenotype. If we look at our therapeutic target, as David has mentioned, we're not the first people to go after LSD1. Big Pharma has had a lot of interest in developing an LSD1 inhibitor. If we look at the two main functions of this enzyme, the first function and its namesake is to demethylate. So that occurs mostly in the oxidase domain, which is shown here in blue. The other important function of LSD1 is that it acts as a scaffolding protein, which means that it recruits other proteins to carry out its full functionality. So that is mostly done by the tower domain, which is shown by this green protruding double helix. What all of our competitors do is they all bind to this FAD cofactor. And you can see that this cofactor is situated in the southern portion of the oxidase domain. Our molecule binds higher up, so not only do we engage the oxidase domain, we also engage the tower domain. So when you compare our molecule to the competition, you see that we're both able to inhibit demethylation, but because we bind higher up in this oxidase domain and engage that tower domain, we're also able to inhibit LSD1's scaffolding properties. So this grants us two main advantages. The first being that by inhibiting LSD1 from associating with these other proteins, we actually promote the degradation of LSD1. And I'll show you why this is so significant for a therapy to do in the next slide. We also have a broader inhibition of these associations, and that's why we have a much stronger reversal of the cancer phenotype compared to the competition, as well as why we have differential activity, meaning we work across a lot more indications than the competition does. And lastly, we are the only reversible binder currently in clinics. So this gives us a much wider therapeutic window, whereas the competition will run into a lot of blood-related toxicities because they're irreversibly binding. We don't see any of those at all. So we have around a 5x greater therapeutic window than the competition. So I'm just going to go into a little bit more detail on why promoting LSD1 degradation is so significant. If we look across different cancers, this data is all done by outside researchers. So if we look at a very hard to treat cancer, triple-negative breast cancer, and we look at the survival of patients, what you see is that patients who have higher LSD1 concentrations have a lower chance of survival. So this curve, it's called the survival curve, every time that line drops, it's because a patient or a group of patients has passed away due to the disease. And you can see the red line drops much faster and it drops all the way to the bottom, meaning that all these patients have passed away around the 50-month mark. If we look at other cancers, we see the same trend where higher LSD1 protein concentrations leads to lower chance of patient survival. So the fact that our molecule can promote the degradation of LSD1 is really significant in terms of impacting the patient survival. The second thing that we do better than the competition is we have a broader inhibition of preventing LSD1 from forming these widespread protein associations. So for example, what this means is that this is cell data. So what we're showing here is the genetic map of a cancer cell. And you can see this cancer cell, specifically it has Ewing sarcoma, that a lot of the genes which allow tumors to grow, so-called tumor permissive genes, they're all turned on. Each of these rows indicates a gene. A lot of the genes that are supposed to keep that cancer cell in check, prevent it from becoming cancerous, these are called tumor suppressive genes, these are all turned off. So that's why we have this huge cancer phenotype happening. When we add in Salarius' molecule, we prevent LSD1 from associating with this complex called NER, and then prevent its demethylation as well. What happens now is you get this complete pattern reversal of the cancer phenotype, where now all those genes that were allowing that cancer cell to become cancerous are turned off, and all the tumor suppressive ones are turned on. So this really highlights how this epigenetic approach is different from those other treatments I touched on. Whereas those other treatments would only affect a few rows, you can see the hundreds of genes that our molecule affects. As I mentioned, this is in cell data. When we translate this into animal data, we get really compelling results, where we see that animals treated with our drug, seclodemstat, their tumor grows significantly slower and then actually completely recedes away. And it's not only a very effective treatment, but it's a very robust treatment and a durable treatment. After seeing complete melting away of the tumors, we stopped treatment for around 6 months, and we didn't see any of those cancer cells. We didn't see any tumor regrowth. So you can see that our survival curve is much better. It's what you want to see, where you have a lot more of the people, or in this case, animals being treated, surviving. If we look at the competitive landscape, as I mentioned before, we are the only reversible inhibitor in clinic. So you can see that there's 5 other irreversible inhibitors, but these all run into that hurdle of having that blood-related toxicity that I briefly touched on before. If we look at what's coming in the drug development timeline, you can see that there's been a shift in focus into developing reversible inhibitors because of the problems that the irreversibles are facing. The good news is that Salarius is years ahead of any of these reversible inhibitors, meaning that we're very well positioned to gain FDA approval before they are. So I want to just focus on our current competition and show how we differentiate ourselves. If we look at GSK's molecule, what we're seeing here is the effectiveness of their molecule across a lot of different cancers. So the higher the line, it indicates more drug concentration is needed in order to induce cancer cell death. You can see that they work relatively well in 2 types of cancer, AML and then small cell lung cancer. That's why they're pursuing these types of cancer in clinic. In the other cancers, they don't work well at all. Even at the highest concentration, they're not seeing any cell death. When you look at our molecule, you can see that those lines are much, much smaller, meaning that we're working at lower concentrations and we're working across all these different cancers shown here on the x-axis. So this just shows how we're able to have more of a potent effect as well as a broader effect across a wide range of cancers. I just want to touch on some animal data that we've shown for the other indications we're going after. So this is in triple-negative breast cancer, a hard-to-treat cancer. It has no therapeutic molecules to target. And what we see as a single agent, we're able to work to significantly impede tumor growth And then when we couple it with another epigenetic inhibitor, we have an even improved response. And moving into clinic, we are going to be looking at seclidemstat in combination with other molecules. So we're hoping to see really great data in these patients as well. And then this is a very hard to treat prostate cancer where the androgen receptor has mutated, and so it's hard to target that with a hormonal therapy. And what we see is that seclidemstat works just as well as the standard of care, which is docetaxel, which is a very harsh chemotherapeutic. So our molecule works just as well at inhibiting tumor growth, but we don't have any of those negative side effects. And once again, this is all as single agent efficacy. So in the future, when we do combination treatments, we're hoping to have a synergistic effect and even a better therapeutic response. And lastly, something of recent news that just came out that we're very excited about is the fact that inhibition of LSD1, or in this case they showed LSD1 ablation, is able to turn cold tumors hot. Immunotherapy is one of the most sought after therapeutic areas within cancer right now, but the huge hurdle that immunotherapies face is that they're ineffective in the majority of patients. And that's because a lot of these patients' tumors are so-called cold tumors. So what happens is that even if you have immunotherapy, you don't really get a response. Here we can see that a checkpoint inhibitor, which is shown by this navy blue line, is basically right on top of the control where there's no inhibition of tumor growth. When you knock out LSD1 and then you add in that same type of therapy, the same checkpoint inhibitor, you see the effect that it has, where there's significantly less tumor growth. It's almost flat. So we're hoping that we're going to be conducting some studies where we can replicate this data and hopefully see the synergy that they show here, where we're able to improve, greatly improve immunotherapies. So I'll turn it back over to David.
Thank you, Daniela. So let's wrap up with timeline and financing. As I mentioned earlier, we are operating under an activated IND. We are currently enrolling patients in the Ewing's phase 1 study. This is the study that the MPCF is funding the majority of our work in. We plan to submit the breast and ovarian and prostate protocols to the FDA by the end of Q3 and hopefully be enrolling patients by the end of 2018. The beauty of these studies is both breast, ovarian, and prostate take advantage of the dose escalation occurring in Ewing's. So there's a lot of synergy going on with this plan. As you can see, by the first half of 2020, we will have data at the maximum tolerated dose across all 5 of the indications that we talked about earlier. Clinical efficacy signals in any of these indications is going to drive the first merger and acquisition window that opens up with this data. So it's very reasonable to expect that as an investor, you have the opportunity to get involved with a clinical stage company with a short-term exit, which is a little atypical for many of the biotechnology investments that are out there. What is it that is represented by the investment opportunity? Our lead investor is Keiretsu Forum. We are raising $7 million. We have about $4 million committed. Pre-money valuation is $15 million. It's a very standard 1x preference plus participation. This means that the Series A investors get their investment back and then participate along with everyone else in distribution of profits. There is an incentive program to invest earlier. So we are currently in the second tranche, moving into the third tranche. You can see the 10% and 6% incentives. The Series A will have a board seat, all the typical protective provisions. This isn't applicable to angelMD, but there is a $25,000 minimum for individual investors. And just to frame this base case that we've been talking about, and it's important to note that this base case has now been validated by 2 separate and independent due diligence processes. A 2020 liquidity event just on Ewing sarcoma could very easily look at about a $400 million value, assuming half up front and half in royalty milestones downstream. A $25,000 investment would return $225,000 at signing and $225,000 over time, which represents an 18 multiple and an internal rate of return of 400%. That 400% is not a typo. That's what happens when someone else is paying for 3/4 of value creation activities in a non-dilutive manner. This leaves upside for related sarcomas, prostate, breast, and ovarian cancers. So this is a very unique opportunity, both from a financing and a speed to exit opportunity. Is deal making happening out there? And the answer is absolutely yes. 20 years ago, big pharma was big R and big D, big research, big development. Today, biotechnology is big R, and big pharma is little r, big D. So when you look at the type of deals that are getting made, obviously the biggest is diversified deals involving multiple disease states, but the single biggest disease state being pursued by pharmaceutical and other acquisitions is oncology, and that's exactly where we are positioning ourselves. We're really confident in our strategy and our plan, and we're already in discussions with a number of companies that are potential acquirers, including Johnson & Johnson, who were nice enough to let us house ourselves here in J Labs in Houston. So, who is already invested? We currently have 3 of the 7 in hand, with another 1.25 to 1.5 soft circle that we expect to come in the next 2 to 4 months. Angel groups include Keiretsu Capital, GPG Ventures, South Coast Angel Network, Houston Angel Network, Rio Grande Valley Angel Network, Texas Angels out in California, and Central Texas Angel Network. We've had a really good mix and have been very successful in being able to fund this small round through high net worth, accredited investor, and angel networks. So we look forward to answering questions, look forward to the angelMD engagement, and look forward to the syndication process. With that, I'll wrap it up and open up the floor to questions. Thank you.
Thanks, David. Appreciate it. It was a great presentation. And I'll note for anyone that's watching this here on the Syndicate page, if you have any questions, along the right-hand side of this page, you'll notice a box that says Ask a Question here. You can either click to email that question and it comes through to me. And whether it's a question about angelMD and our syndication process, or specifically about Salarius, I will either get it answered by myself or send it over to the Salarius team for them to answer it for you. If you have any other questions about the syndication process, what our minimums are, and how the terms work, you can find that information here on the About Us page of angelMD, or again, feel free to give me a call anytime. As for questions on this particular one, Dan, I'll open it up to you first if you have any.
Yeah, thanks, John. So my first question, David, I wanted to start with the exit slide that you had, and obviously that's extremely compelling. And I wanted just to ask if you could clarify a couple of things for me on that. I understood you to say that the 200 upfront and the 200 back-end potential scenario, that is just associated with Ewing sarcoma. So 2 questions on that. Should there be good indications for the hormonal cancers? What do you just imagine that that could look like from a potential increase in value? The other question is, what happens if you don't have the indication that you're expecting in phase 1 that may lead to that quicker exit at the end of phase 1? What happens at that point?
So let's take those one at a time, Dan. In the event that we have Ewing sarcoma and another indication, I would expect total deal value to increase. With the other indications, hormonal cancer, requiring the more traditional phase 1, phase 2, phase 3 development program, along with comparator studies where they have to show equal or improved efficacy with competition, I would expect maybe a slightly increased upfront payment, but a much bigger back-end payment. And that's typically due to the economics of the deal structure. Big Pharma has the money to put in, but they're also going to want to time the payout of the economics to more align with when the revenue starts coming in.
Is it possible that you could structure a deal just for the Ewing sarcoma and then separately and differently the hormonals?
Dan, I get that question a lot.
Yeah.
And I've spent over 22 years in Big Pharma. I've been involved with a lot of different deals. I generally have not seen deals where indications are licensed to different organizations, especially in today's environment with the collection of safety data, but more importantly, pricing considerations.
Mm-hmm.
You really want to keep your indications together and be able to promote a molecule. But what would be very likely is the possibility of licensing or selling the rights to the drug across different geographical regions to allow experts and powerhouses in the U.S., a powerhouse in Europe, and a powerhouse in Asia to optimize the value of seclidemstat. I think that's the more likely scenario.
Right. And at what point do you believe you'll have early indication as to whether this phase 1 will result in that, or is that really at the conclusion of the study?
Dan, that's another question I get quite often, so thanks for asking. It's really hard to tell. The reason is patients can respond differently.
Mm-hmm.
It is possible, and for the sake of patients, I hope this happens, that we can see the beginnings of a clinical response in month 2 or 3 of the first 2 or 3 patients. That would give us data in early 2019. Now, that might be a signal. And a signal is great, but then you're going to want to see, well, how often is that going to repeat as we enroll more patients? So I think the realistic answer to your question is, it's certainly possible to see something early on, maybe in Q1 of 2019, but I think a more realistic answer is probably the end of 2019, early 2020. Now, let me come back to the second question that you asked early on, and that is, how can the data play out? And what happens if you don't see what you're hoping to see early on? And the answer is, that's not uncommon in drug development. And drug development is a learning process. But what I feel very confident in is the strength of our preclinical package is number one, that all sends us down in the direction that we're going to see something. It's just hard to imagine that with all of the biologic activity we've seen in our animal models that we're not going to see something in humans. The second part of the question, or the answer to the question, relates to diversification. We're not a one-trick pony, one asset going after one indication. We have one asset going after 5 indications, which is a very diversified portfolio, in addition to some very exciting immunology opportunity that Daniela touched on. So to answer your question, I think one thing I do know for sure is we're going to learn things we didn't know before, but I think we have enough shots on goal that this is a nice way to play an investment.
David, I realize it's a data point of 1, but the compassionate use study is one that certainly gives some promising insight in the potential of the drug. Can you elaborate on that a bit, and what about that study gives you the first? So
the compassionate use study was it was a tremendous opportunity for our patient. And unfortunately, this was an end-stage patient who was entering hospice. Mm-hmm. But he was so passionate about not only his potential for treatment but also helping others. He really allowed us to learn a lot. So the key things that we took away from that compassionate use study is, one, we know we are able to get blood. We can get drug into plasma at therapeutic levels. That's one of the key unknowns. Until you actually start treating a patient, you don't know if you can get drug at therapeutic levels into the bloodstream, and we can. So that's one big de-risk factor. Two was the lack of toxicity that we saw, another big learning. And then three, when you look at the clinical study report, the treating physician noticed a regression of primary tumors, a shrinking of primary tumors, which again, to your point, how early could you see something? Well, it's just one data point. We didn't get a chance to track the patient because it was compassionate use, but the fact that we can get drug into the blood at therapeutic levels, that we didn't see toxicity, especially didn't see toxicity of some of the other irreversible inhibitors we're seeing, and that there was at least some indication that we were impacting primary tumors, it's very exciting.
Yeah. So Danielle, I have a question for you. The slide that you showed where there was a reduction in the LSD1 levels and in the increased survival there. What do you imagine? What explains the difference in the LSD1 levels in those studies?
Yeah, so that slide showed that higher LSD1 levels within the tumor leads to decreased chance of that patient surviving, right? That's why.
Right.
And what's likely happening is that LSD1 has a necessary healthy function in stem cell development. So what's likely happening is that these cancer cells are hijacking this LSD1. By them having higher levels, they're acting now as these cancer stem cells, which are well known to be harder to treat. They're well known to be more aggressive, and that's why you see a decrease in patient survival when there's higher LSD1.
Okay.
So the fact that we can promote the degradation means that we can prevent these stem cells from becoming like cancerous stem cells and hopefully reduce the aggressiveness of these cells, make them easier to treat. In combination with then having the therapeutic effect that we have, it should have the synergistic effect. That's what we hope to see.
Right. That's it for me. Thanks.
Thanks, Dan. I have one quick question for you, David, regarding VC firms. I know a lot of people get concerned, especially around these types of investments that could take a while and cost a lot of money, that a VC firm might come in, beat down the earlier investors and restrict some of the access to information and everything else you typically share. Can you talk about how Salarius kind of fits in with VC firms and how you see them potentially or not potentially playing a part in the company going forward?
Well, John, we've talked about that a lot at Salarius, and so I'll start with where we are and where we might go. Where we are is that with the tremendous support from the state of Texas and support from the National Pediatric Cancer Foundation and from the University of Utah, we are currently not pursuing venture capital investors, and we're in a really unique position where typically a company entering the clinic needs $25 to $30 million, and we're no exception. Our number is 28, but instead of having to raise that full 28, I only need to raise 7, which means that we're able to take a little bit longer runway, but raise our 7, as I mentioned earlier, from high net worth individuals, family offices, accredited investors, and angel funds. We're right on path where we thought we would be, and the benefit is we're able to, for the foreseeable future and through the completion of our phase 1 studies, really focus on what's best for the patient, for the company, and our individual investors without the involvement of venture capitalists. Now, the second part to your question is, what does the future look like? Well, there's a lot of opportunity out there, and it's all going to be data-driven, but we think we have a nice program put together. We think we're going to have meaningful clinical data coming out at the end of 2019 and early 2020. And we think that sets us up for a variety of options. You know, one is an outright acquisition. One is a process called reverse merger, where a public company who needs to bring in assets could merge with us, and we immediately go public, creating an liquidity event for investors. The initial public offering market is pretty hot right now. I don't know what it'll look like in a few years, and that's not always my favorite path forward, but it's one that's out there. And then failing that, you know, you always have the opportunity of going back to venture capitalists. But at that point in time, I have to think we're going to be in a position of strength with a lot of data in our hip pocket, and we'll be in a much better position to dictate terms then than we are right now. So wrapping up your question, I'd say this is, again, a unique opportunity for angel investors, angel funds, and individual investors to get on board with a clinical stage biotechnology company without the influence of venture capitalists for the foreseeable future.
Thanks, David. I appreciate it. And that about wraps up what we have for today's presentation. I'll make also a note that we do have some Syndicate events coming up for Salarius, both in the Houston area and in Dallas. Those events and their details can be found on the events page here on angelMD. And they are open to angelMD members for attendance. The investment minimum for this syndicate is $10,000. We can accept funds via self-directed retirement accounts. Should you have an interest in doing so, please reach out to me via that Ask a Question box about your retirement funds, and I can get you started on that. It can be a lengthy process to get that approved, so please do not delay in that regard. You must be an accredited investor in order to meet the standards necessary to invest in angelMD syndicates. We do ask that you go through a third-party verification service that will run you through uploading either tax returns or items that can prove net worth. If you have any other questions, you know where to ask. Otherwise, thanks so much for joining us today, and we look forward to you investing on angelMD.